What are opsins in optogenetics? Imagine being able to turn neurons on or off just by shining a light. Sounds like science fiction, right? Well, in the world of neurotechnology, this is a real and powerful technique called optogenetics. At the heart of this method are special proteins known as opsins. These proteins are sensitive to light and act like tiny switches inside cells. When light hits them, they change shape and trigger a response in the cell. This response can be to activate or inhibit the neuron, giving scientists precise control over brain activity. Opsins are a family of proteins that respond to light by changing their shape. They need a molecule called retinal, which comes from vitamin A, to work. When retinal absorbs light, it shifts shape, causing the opsin to do something. It might open an ion channel, letting charged particles flow into or out of the cell, or it might activate an ion pump that moves ions across the cell membrane. This movement of ions changes the electrical state of the neuron, making it more or less likely to fire. There are two main types of opsins used in optogenetics. The first type is microbial opsins, which come from microorganisms like bacteria, algae, and fungi. These are popular because they are simple proteins that respond quickly to light. Examples include channelrhodopsins, which open ion channels to activate neurons, and halorhodopsins, which pump ions to inhibit activity. The second type is animal opsins, found in higher animals, including humans. These are involved in vision and biological clocks, but are less common in optogenetics because they work through complex signaling pathways. Scientists insert microbial opsins into specific neurons using genetic techniques. Once inside, shining light on these neurons makes them respond in a controlled way. This allows researchers to study how different parts of the brain work or to develop new treatments for neurological conditions. Since opsins need retinal to function, many mammals already have enough retinal in their brains, so no extra steps are needed. However, some animals like fruit flies need dietary retinal to activate these proteins. In neural implants and neurostimulation devices, opsins act as biological switches that turn light signals into electrical signals inside neurons. This ability is key for developing advanced brain-computer interfaces that can control neural activity with light. Such technology could someday help restore vision in people with retinal diseases or treat brain disorders by targeting specific neural circuits with high precision. To sum up, opsins are light-sensitive proteins that make optogenetics possible. They control neuron activity by responding to light and changing ion flow across cell membranes. This makes them essential tools in neurotechnology, opening new doors for research and medical treatments.